Hello and welcome everyone to day 2, session 7, the last session of AI Confluence, and it is Neoepitope prediction and prioritization at a whole genome scale. I am Anushwa Chatterjee and I chiefly work in the domains of applied AI in genomics, transcriptomics, cancer biology, and infectious disease biology research. Our agenda for today is chiefly what are exactly neoepitopes, their applications, their various importance, challenges in the traditional candidate neoepitope design, and how we have used and implemented AI to solve through majority of the bottlenecks and challenges in epitope design and clinical prioritization. Quantify solution in the development of an integrated, high throughput, multimodal, automated platform for new epitope discovery, prediction, and final filtration, the results and discussion of our models, and the business impacts and key takeaways. So, where are exactly neoepitopes used? And that is cancer. So, before we go into neoepitopes and their application and their custom development, let us take a brief walkthrough over the disease, cancer. So, cancer is essentially a malignant or a neoplastic transformation of a tumor. They can be solid when they are called tumor or hematologic when they are blood based, like leukemia. So that is essentially when a group of cells start dividing uncontrollably and form certain lumps outside or mainly inside the body, and then certain cells from this lump slough off and migrate to other parts of the body via a process called metastasis, and they travel through either bloodstream or lymphatic system, and they go and lodge there and form secondary tumors. So that's how cancer gradually spreads throughout the body and eats up the body from inside. So they're essentially classified on where the primary tumor originates and the type of cell, like bone marrow, connective tissue, skin tissue, melanocytes, etc., in which part of the body, which type of tissue it affects. Currently, the traditional treatment procedures include surgery, surgical resection of the tumor, chemotherapeutic drugs like cisplatin and all. Then there is radiation therapy where gamma radiation is inflicted upon the tumor in order to slow down the rapid growth of cells and slow down the metastasis. Immunotherapy, which is a more of new age revolutionary treatment where the antibodies, that is, the antibodies engineered against a specific target, are harnessed from the patient's body and used against that particular tumor, and they help to activate the immune system against these specific malignant cells and destroy the cells from inside. Various kinds of targeted therapies, which are developing as a host of precision medicine and personalized targeted therapies. And then we have hormone therapy, which is limited to a particular type of hormone sensitive cancers, like in breast cancer or prostate cancer. So, our main problem statement here is the traditional treatments that the patients undergo, like chemotherapeutic treatments, radiation therapy, hormone therapy, often they have been the backbone of oncology for decades, and patients have resorted to them as the first line of standard care or treatment. However, they have some severe limitations and drawbacks, including they're highly non-specific and they target both the cancer cells as well as the healthy cells of the body, causing severe toxicity, very high relapse rate, that is, chances of cancer coming back again even after the patient has fully recovered, poor efficacy against certain types of resistant or recalcitrant tumors. So these challenges highlight us the urgent need to go from a paradigm shift from the generalized approaches of treatment and therapy towards a more targeted approach where we leverage the patient's immune system to specifically target and attack the cancer cells, the malignant cells, sparing the healthy cells, and thus help the patient to recover fully and successfully without any of the dangerous side effects or the relapse rate. So, Targeted therapy is a major challenge in cancer, but with development of new epitopes and the evolution of neoepitope based personalized precision immunotherapy, we seek to overcome this hounding problem. So, introduction to neoepitopes and its importance. Now, as we talk about this urgent, necessary shift in the treatment paradigm from the generalized approach towards a more customized, tailored, specified approach in personalized precision therapy, we talk about what are neoepitopes and how exactly these neoepitopes will help us to do this paradigm shift. So neoepitopes are essentially very tumor specific and tumor exclusive antigens which arise from certain mutations in the DNA of these cancer cells. So they are like the special mutated ID badges which are present on the surface of these cancer cells and not on the healthy cells. So these are presented by certain molecules which we called major histocompatibility complex MHC molecules, and these molecules show these mutated ID badges to the T cells, which are a part of the immune system. So the immune system recognizes this foreign, non-self enemy badges and immediately attacks it with a much more heightened, accelerated and stronger response, and also offer often a lifetime, a very long-term immune memory protection which drastically reduces the chances of cancer relapse. Now, combination of such kind of targeted therapy with checkpoint therapy, adoptive cell therapies, and CAR-T therapy has drastically improved outcomes, progression-free survival, and disease-free survival for patients, and even reduced tumor resistance and recurrence, thereby greatly improving the quality of life of cancer patients. Now, this diagram here gives us an illustrative workflow of actually how these new epitopes are processed inside the cell, and then they are carried by these small protein molecules, which are called MHC, to the surface of the cell, where they are displayed to the CD8 plus T cell, which are basically the killer cells of the immune system, which come bind with their TCR, that is a T cell receptor, to this binary complex of the MHC and the neoepitope together, and this elicits a complex reaction which activates the CD8 killer T cell and the immune system, and they attack and kill the specific malignant cell. So this is exactly how the neoepitopes function and what they exactly are. Now, there are several bottlenecks in candidate neoepitope design. It is extremely difficult to identify those few specific targeted neoantigens which can particularly trigger a very potent and accelerated and heightened immune response that can actually kill off the malignant cells, and they are very heterogeneous. They have a high degree of genetic heterogeneity and a biological complexity due to the varying genetic makeup of a patient, and there is a very low yield of actually effective neoepitope targets, especially in those kinds of cancers like breast cancer or lung cancer or prostate cancer, where there is a low tumor mutational burden, especially in their early stages. And then we have to ensure effective presentation and the binding affinity and binding stability of these protein neoepitopes to the MHC molecule and to the T cell receptor to ensure the proper interaction that can thereby trigger the signaling cascade which activates the immune system. And then finally, we have to continuously improve their clinical relevance and their prediction accuracy over time. So these challenges we have found through our research can be solved and even augmented with AI-powered new epitope filtration, prediction, and prioritization. So we employ specialized deep learning models to predict and rank high potential, highly immunogenic epitopes from large genomic, whole genome, whole exome, RNA-based data sets of patients. We process and these models employ vast patient-specific data to curate these highly specific neoepitopes and create these specific personalized predictions. And then we have developed a model to intelligently score and prioritize candidates on several various biological key metrics like MHC binding stability to maximize the hit rate, then accurately simulate these complex biological signaling cascades and molecular mechanisms to find the correct candidate neoepitope targets with the highest affinity. And then we finally use T cell receptor modeling simulations using adaptive learning from clinical response data to fine tune these models, fine tune the LLM based models for enhancing and augmenting the identification of these potent peptides. So, owning to the various multifaceted applications of neoepitopes, they offer a whole new dimension to the new developing area of precision medicine, of precision oncology, and they provide very robust, effective, and clinically actionable therapeutic targets while minimizing the off-target toxicities and the various side effects and disadvantages which are associated with traditional cancer therapies. One notable alternative is that the recent advances in the highly immunogenic and tumor-specific neoepitope targeted therapies like cancer vaccines, where we employ mRNA or these short peptide-based vaccines to prime or just like give a jolt to the immune system, particularly helpful in the type of cancer where there is a very immunologically cold or we say non-responsive environment around the tumor, which does not allow the immune cells to come and attack the cancer cells under normal conditions. But such neoepitope primed cancer vaccines can help these cells to infiltrate the tumor and attack the cancer cells specifically. Then we have certain cell-based therapies like CAR T therapies, where the T cells are engineered to target, they're genetically engineered to target the specific neoepitopes on the cancer cells. Then we have specialized adoptive cell therapies and precision oncology-based therapies like neoepitope profiling, HLA typing, ctDNA profiling to guide therapy selection, to to effectively guide the correct disease prognosis, diagnosis, and treatment outcomes of cancer patients. So now we would give you a glimpse into the current ongoing work, our workflows and architectures. So we at Quantiphi have developed a multimodal, high throughput, fully integrated, automated platform for new epitope discovery, prediction, and prioritization from patient-specific whole genome sequencing or WGS data. So as you can see in this illustrative workflow, we have depicted here a high-level understanding of the architecture that we have currently implemented to filter out the best candidate neoepitopes and then proceed forward for the best for and then proceed forward for experimentation, in vitro experimentation, to derive the most suited clinically actionable targets for these precision oncology therapies. First, we take the whole genome sequencing or the RNA sequencing data of patients. So here we have taken an advanced progressive breast cancer cell line data, and we have processed it using the NVIDIA Clara Parabricks platform. So here is a quick chart, a quick workflow of how we have processed the raw data. We have taken the whole genome FASTQ data, indexed the files, performed quality controls and checks, done a quick reference genome indexing. That is, we have compared the cancer data, that is the data with the mutations, with the healthy data to see where we have the mutation, at which position, at which genes, done various like read mapping, and then variant calling using mutate2, and finally with the filtration, which was aligned with the GATK best practices and annotation, we got this analysis ready variant call format VCF file, with which we have progressed to the next steps. So here essentially we have filtered out those particular mutations, those particular positions where cancer-specific gene changes, amino acid changes, base nucleotide changes are there. We have filtered out those positions. We have created a file with all these filtered raw mutations. We have done a functional annotation to it. We have given it a functional significance and a disease association, like how these mutation or base changes are associated with cancer or associated with a particular disease. Then we have determined the specific MHC molecule. The MHC molecule, that is the protein which was carrying the new epitope to the surface of the cancer cell for presentation. We have determined what particular ID of the MHC would be needed here. For this, we have used a bioinformatic tool called Optitype. Then we have used a software, an integrated software suite, PVAC, and integrated it into our multimodal pipeline, where we have used it to extract the whole plethora of the tumor associated antigenic epitope peptides. Like these are 9 bases, 10 bases, or 12 bases long, which we call 9-mer or 12-mer long antigenic peptides. So we get about like 10,000 to 15,000 of peptides extracted from this raw mutation data. And then we begin a series of steps to filter most the robust, the most precise, and the most clinically actionable new epitopes from these 10,000 to 15,000 epitopes. So we do various binding affinity studies with various deep learning models to see which of them specifically bind with the maximum binding affinity, maximum binding stability, and very high presentation potential. Then we have developed here a customized model wherein we have leveraged ESM2, the protein large language model, and combined it with PSSM matrices and developed a consensus scoring method to score the final ranked neoepitopes on the basis of their immunogenicity, like how good these epitopes would be to elicit and trigger the immune system to attack the cancer cell. That is something, the property we call immunogenicity, and as well as the stability with which they can bind to the MHC molecule and the T cell receptor. So based on this consensus score, we rank and filter these neoepitope peptides, and then as the final litmus test, we perform binding stability and binding affinity studies with the T cell receptor. And that's how we come to a handful, say like 5 to 10 neoepitopes, which are our best candidates for, uh, which are best clinically actionable candidates to be used for further downstream applications like CAR-T therapies, cancer vaccines, and other neoepitope-based adoptive cell therapies. So the very core of our pipeline is this dual stream architecture for immunogenicity prediction that synergistically combines two information sources, from the SM2 protein large language model and the PSSM matrix, to generate a combined consensus score to filter and rank new epitopes on the basis of their immunogenicity and binding fitness. So one information stream here comprises of the ESM2, which has been trained on large data sets from IDB and other publicly available data sets to learn the contextual embeddings and the complex sequence motifs and the non-local relationships of these immunogenic peptides. And then the other information stream is from position-specific scoring matrices, PSSM, which gives us a score that represents established peptide MHC binding fitness. Now, the output from both the streams are then fed into an attention-based MLP head, and that generates a single consensus score. So the fusion model has given enhanced from preliminary evaluations. We have seen this fusion hybrid model gives us enhanced predictive performance over the ensemble based or CNN based single modality approaches, which are currently there to predict such neoepitopeptide immunogenicity. And this hybrid framework, it gives us the best of both worlds, as in it combines the statistical power of established binding matrices with the deep contextual learning of modern transformer-based large language models to make an accurate, informed, and robust prediction on peptide immunogenicity. From here, this ranked and filtered epitope list is finally fed to the T cell receptor binding and simulation deep learning models. We have employed the trap TCR here to generate binding affinity scores, which would finally determine the best candidate new epitope for in vitro experimentation. Now, coming to the last stage of our discussion are the results of our model evaluations. So initially, we had performed the variant calling and annotation with our Parabricks suite and the VP annotation software. So from here, we get a complete list of all the candidate antigenic epitopes with the chromosome position, at exactly which position the mutation has occurred, what kind of mutation, whether it's an exonic, it has occurred in a exon that is a readable region or intron non-readable region, which particular gene it has affected, and what kind of mutation it is. And then we have various metrics associated with amino acid change. From this table, we get an idea of the disease annotation or the functional annotation, what kind of gene, what kind of mutation is there in our variant data, in our mutation data, so that from here onwards we can go and filter out the most prominent genes and the most prominent mutations, and can use them to finally list out our new epitopes. So this is a glimpse of a few extracted peptides that we got from our PVAC-seq results. That is, we had given the variant call format file to the PVAC-seq for extraction of all the candidate 9-mer, 10-mer, and 12-mer peptide sequences. So as you can see, we have got the hits of the best peptide sequences, the associated allele along with it. So here we get an allele code B45. Like this, we have several other candidate alleles which bind. So this is the HLA or the MHC molecule, which specifically binds to this peptide. And then we have their binding affinity, their percentile scores, which I would be explaining in the next slide. So yeah, we get this ranked list of filtered epitopes, which we then feed to several deep learning models. In this case, we have used MHC Flurry and NetMHC Pan for determination of the binding affinity and binding stability of these epitopes to the MHC molecule, and that is by the binding affinity IC50 scores. So these IC50, so the scores essentially tells us that how much or like this concentration of this peptide would be required to displace the reference or the naturally occurring binding peptide to this molecule. So the lower the score, the more is the binding affinity, the more is the stability. And the net MHC pan here gives us the elution ligand scores, which is essentially an estimate of what is the probability of this particular peptide. It's going to go through the natural biological processes and is going to be presented on the surface of the cancer cell to the T cell receptor. So we get these scores here. The elution ligand scores are essentially the presentation probability scores, and then what kind of mutation it is, what kind of displacement has occurred here. It's a missense kind of mutation, and then the median empty percentile. So based on evaluation of all these scores, we are able to select the best epitope candidates. As you can see, we have shortlisted here 5 epitopes which has matched our baseline, our threshold criteria and characteristics. So for the ESM2 PSSM hybrid core model, we are still more of in the testing and preliminary evaluation stages. So these are the key performance metrics of the initial baseline model that we are currently testing. So as you can see here that these are the preliminary results, the key performance metrics of the initial baseline hybrid ESM2 PSSM model that we are working on. So we are currently still in the testing and optimization phase of this model. So as you can see from these performance metrics, it is yielding a low efficiency result for now, and we are getting more of a highly optimistic results with a greater number of false positives, which is leaning our model towards the like towards more of a higher number of positive results. So yeah, we are working on the feature feeding inputs to the model because the most significant gains always comes from the right amount of features and from improving the features which one gives to the model. So here we are currently working on developing the HLA matrix, HLA allele matrix. So they serve as categorical features, and we want to create a like deep a dense vector representation of the HLA allele matrix, and then concatenate that vector with the pooled output from the cross attention block prior to the final classification layer. And then once we concatenate it, it finally goes to the final attention based classifier, which then ranks, which then like classifies between the immunogenic and non-immunogenic peptides or neoepitopes. And apart from this, there are a few other future action items which we like want to work. So apart from this, we are also working on optimizing the loss function. Currently, we're using a binary cross entropy loss. However, that treats all the samples equally. So maybe substituting the BCE loss with a focal loss to dynamically weigh upon the contribution of the missampled peptides. So that is also another key metric that we are considering. In addition to that, we are also working upon adoption of a dynamic learning rate scheduler, dynamic learning rate, like a reducer around plateau to ensure a more stable and optimal convergence during training, and like avoid like get more fine-grained convergence and avoid more suboptimal local minimas. Also, in addition, we are working on the adoption of a per amino acid residue based masking strategy and training of the PSSM matrix on a more balanced data set. So currently it's trained on only the positively immunogenic peptides, which is probably giving the more optimistic results. So we wanted to train it on this balanced data set and then incorporate it to the cross attention layer to make the final attention based classification. So these are the future action items that we are currently working upon and intend to do to improve the model performance metrics. And we firmly believe that it will make a very robust, high performing, one of its kind novel model that will be able to give us a very good consensus score of the peptide immunogenicity, which can then be used to rank the peptides and choose the best candidates. So coming to the last part of our slide, that is the key impacts, the business impact of our novel invention. So yeah, this this Neoepitope-based pipelines, it comes under the area of AI-driven personalized immunomics. So yeah, this leverages tumor-specific neoepitopes to drive the upcoming next generation based targeted, precise, and highly successful cancer therapies like CAR-T, adoptive cell therapies, and cancer vaccines, creating a more effective new new paradigm shift of good successful treatments. It opens the billion-dollar markets in immuno-oncology, personalized treatments, and precision oncology, comparable to the recent checkpoint inhibitor markets. It provides a competitive edge in developing personalized, tailored approaches for patients because we have developed this entire pipeline and this entire ranking and filtration of the peptides, taking an individual patient's genomic data. So these peptides are customized to each individual's genetic makeup and to experience to and because this is a GPU accelerated high throughput pipeline. So yeah, we would be able to upscale it to many more, to thousands, to like get thousands of new epitopes, thousands of peptides together to get like faster R&D cycles, and definitely for precision medicine adoption, and like ultimately help in advancing the next generation of revolutionary cancer therapies like CAR-T. So yeah, ultimately we get this ranked list of new epitopes, and we can provide them to the experimentation labs for clinical trials, for in vitro experimentation, for testing, for human and mice trials, so that we can finally get those clinically actionable, robust, human safe targets, which can then be employed for the new era of revolutionary precision oncology therapies. Thank you.
Great. Thank you so much, Anusuya, for your informative talk. We are really over time, but maybe let's just quickly take one question from the audience.
Sure.
Let me just read out. So, is there any specific reason to deploy a transformer PSSM approach based hybrid model to rank the epitopes?
Yeah, that's a really insightful and interesting question. So, based on a literature review of all the previous models that were out there for the neoepitope prediction, for development of these vaccines and therapies, we saw that they are mainly based around the CNN-based or the LSTM-based approaches, which are not exactly that efficient to. So, they have a limited biological context window. They're not able to capture the sequential information, and they have very inefficient feature extraction. So we saw when we tested these ESM2, the pre-trained ESM2 transformer, coupled with the PSSM matrices, we were able to capture large functional contextual windows and capture local sequence embeddings, local sequence motifs, as well as introduce the evolutionary and positional information along with it. So that helped us together to give a synergistic, context-aware scores, which captured the actual nuanced biological, structural, and functional understanding of these neoepitope peptides. So, yeah, that is the main reason we employed this novel approach here.
All right. Thank you so much, Anusuya.